Okay, so here we're going to talk about viruses that infect eukaryotic cells. So whether it be mammals or plants or other things that are eukaryotes. But a virus, it's important to remember that a virus is not a cell. So when we think about the characteristics of a cell, like all cells have a cell membrane, all cells have a cytoplasm and ribosomes and DNA. A virus does not meet those qualifications. So when we look at, well, what is a virus? A virus, its genetic material, its nucleic acid, can either be DNA, RNA, like a single strand of RNA, or it can be both. And sometimes it can even have double-stranded RNA. It doesn't follow the same rules of like how eukaryotes have linear chromosomes and prokaryotes have circular. It doesn't necessarily follow those rules. And instead of being surrounded by a cell membrane, it's surrounded by a protein coat. So this protein coat could have certain like glycoproteins on its surface. So when we talk about these proteins on the surface of a virus, this is one of the ways that our immune systems can identify that this virus does not belong inside of our bodies. Now, each of these protein coats, I believe on my next, hold on, slide. So, well, I guess I'll get there. Another thing that might occur in some viruses is they can pick up a like membranous or like a lipid bilayer envelope from a host cell. So after they replicate and they are exiting a host cell, they can take some of that host's cell membrane with them, kind of. So here's words for people who like to read. Now, a DNA is called a virus is called a DNA virus or an RNA virus, depending on the type of nucleic acid it has. So I'm actually recording this right now during the time of COVID-19, and that is a coronavirus. That is an RNA virus. Now, that name coronavirus comes from the fact that when you look at it underneath a microscope, it looks like a crown. So, like, because corona means crown. And so, when you look at like these proteins around the surface, underneath the microscope, that coronavirus would look like a crown based on those proteins on the surface of that protein coat. Okay, so when we look at this protein coat, it is more officially called a capsid. And so this comes in a bunch of different shapes and sizes. So if this is a virus that is infecting a plant, it might be more like a cylinder shape. And then we'll see in another video about viruses that infect bacteria, and those look like cute little robots. And so this protein shell or this capsid does come in different shapes. And there are also maybe these glycoproteins, which are going to be proteins on the surface of the cell. And we talked about some of them have a like a envelope, a cell membrane envelope around the virus that they derived from that host cell. So that does kind of make it easier to like enter into nearby host cells inside the body. So here, if we look at this like Google image kind of, you can see the nucleic acids, whether it's DNA or RNA or both, is on the inside. And then you have that protein coat or capsid. And then those spikes that you see are those glycoproteins. Each virus has their own unique glycoproteins. And this is how our immune system works when we identify a foreign like pathogen inside our bodies. And when we talk about the like memory cells, like when you develop immunity to a virus, it relies on the fact that those proteins aren't going to change. So, for example, when you get the flu, you develop immunity to that flu virus, but the flu virus is constantly mutating. And if its nucleic acid mutates, well, then that's going to cause different proteins to be made. And therefore, those proteins will be different on the surface. Those glycoproteins will be different. And now it's a new flu. And therefore, you no longer have immunity against a new virus. So when we develop vaccines for viruses, we are counting on them not mutating, kind of. So viruses, though, because they are not cells, they can only replicate within a host cell. So when we look at, here's a host cell. Now, here we have a virus. You can see I have that purple represents the capsid or the protein coat around it. And those green squiggly things represent the nucleic acids, whether it's DNA or RNA. So once it enters into a host cell, now in reality, viruses have different ways of entering cells. They could enter through like one of our like receptor proteins or something. And so these viruses could have certain enzymes that can kind of like very simply like break into a cell. But I believe like the real in-depth discussion about how a virus enters a cell is beyond the scope of AP Bio. So if you major in like immunology or something in college, I'm sure you'll learn it in more detail. But anyway, so now the basics of how a virus replicates within host cells. So it's going to like disaggregate, like break apart within that host cell. And this is where and why it needs a host. Because viruses don't have their own ribosomes, they need the host cell's ribosomes. And so the viral DNA or viral RNA contains genes that are going to have the directions on how to build a new capsid and how to build new proteins for new viruses to spread. However, so if this is viral DNA, for example, the host cells, like like RNA transcriptase, is that the name? RNA polymerase, oh my gosh, so sorry, will then transcribe the DNA, the viral DNA, make messenger RNA that then codes for new viral proteins. And then we could also do replication of that nucleic acid to make more copies of it. And then once you have the nucleic acids replicated and you have the new proteins that will make that capsid and glycoproteins replicated, now those viral components will aggregate within the cell and self-assemble. Now, once they self-assemble, now they can exit the cell and go and infect other cells. So this is the step when they exit the cell where they may take part of that host's cell membrane with them. So here's like a textbook image. It's a little bit prettier than mine, but you could always pause a video and study it or just open a book or go to Google Images. Okay. Now we do have some viruses that are called RNA retroviruses. Oh, I should mention since this is coronavirus time, the coronavirus is an RNA virus that uses the RNA, and that RNA has the genes in order to make more copies of its capsid and replicate that way. So yeah, in that messenger or in the RNA of the coronavirus, it'll just make copies of the RNA. And then in that RNA has the genes to produce the proteins that will form the capsid and all those glycoproteins. Okay. Now, when we look at another class of RNA viruses, these are called retroviruses. So retroviruses are pretty like tricky and super interesting. So a retrovirus, just like the word retro implies, like retro is like back in time or before. So in a retrovirus, they actually will work backwards, where a retrovirus carries its own enzyme called reverse transcriptase. That's why I was messing up that word earlier. But when you think about reverse transcriptase, it's exactly like it sounds. It's doing transcription in reverse. So if you think, well, transcription starts with DNA, and then that DNA is copied into RNA, well, now you're going to start with RNA and work backwards to make DNA. And so one of the most like famous examples of a retrovirus is HIV. So let's go ahead and see how a retrovirus works. So here you can kind of see the glycoproteins around that virus. You can see the pinkish color RNA, as well as those yellow circles are the reverse transcriptase. Okay, so let's go ahead and see how this works. So here you have an HIV virus entering a cell. Now, if you've studied the immune system, HIV actually enters helper T cells. And there's a particular protein receptor on the helper T cell, and that is the receptor that HIV enters through. So there are a handful of people of European descent. I forget, I don't remember exactly which region or which country of Europe, where there was a mutation during the Black Plague that their helper T cells were missing that receptor protein. And it helped them survive the plague. So by natural selection, those people were selected for and were more likely to survive the plague. And if they survived, that means they lived long enough to reproduce and have offspring. And then their offspring inherited that mutation where they also were missing that receptor protein. And because it was a high fitness trait during that particular environmental time period, that phenotype or genotype or both increased in percentage or increased in frequency in the gene pool. Well, today, even though it hasn't continually been selected for, today there are a handful of descendants of these people who their helper T cells are do have that mutation where they're missing that receptor protein, and therefore they're immune to HIV. So pretty cool. Anyway, so HIV enters the host cell, and what's going to happen next is it will disassemble. Its protein coat will come apart. And now it has this viral RNA. And this is where we're going to work backwards and do reverse transcription. So in reverse transcription, the enzyme reverse transcriptase will read the code in RNA base pairing DNA nucleotides. So now you'll get an RNA DNA hybrid. And from there, we can take one strand of the DNA, and because DNA has like a complementary like base pairing rules, if you know one strand of DNA, you can find its complement. Now, this is the part that makes HIV so devastating, is that now that viral DNA will incorporate itself into the host's chromosomes. So now you have this like pro-virus section. You have now that viral DNA forever integrated into that chromosomal DNA. And now once that gene is inserted into it, that viral DNA can now go through protein synthesis. So then you'll have transcription where RNA will be produced, and then from that RNA, proteins by the host's ribosomes. And then those HIV viruses will like self-assemble and then exit the host cell and go and infect other cells. So retroviruses are different because they rely on those reverse transcriptase. So this is different than like the coronavirus example I used. Like the coronavirus doesn't use reverse transcriptase. Okay, and so that is going to end my video on viruses that infect eukaryotes. And then my next video will look at viruses that infect prokaryotes. Good job.